So, good morning, everyone. I'm Yasmin Khan, and on behalf of the Department of Life Sciences, Mumbai College, I welcome you all to this webinar on COVID-19 and immunology perspective. We are grateful to Dr. Satyajit Rath for accepting our invitation. As per his suggestion, we have kept the format of this session as an interaction between him and the participants instead of a formal talk. So students, please take advantage and get all your queries answered and doubts cleared. We acknowledge the support of DBT Star College scheme that has been awarded to our college, and in keeping with the spirit of collaboration, which is a mandate of this award, all the participants are students and faculty from the Department of Life Sciences from the other Mumbai University colleges, and our own students from various biological sciences departments. I would like to thank the heads of all these departments for their support, because very promptly in response to the request, they had sent in lists of students almost immediately. I will now invite my colleague, Dr. Bhavna Daswani, who has done all the behind-the-scenes organization for this webinar, to formally introduce our guest, Dr. Satyajit Rath.
Thank you, ma'am. And a very good morning and a very warm welcome to everyone present here. It gives me immense pleasure to introduce our expert for today's session, Dr. Satyajit Rath. Dr. Satyajit has been trained as a physician and a pathologist in Pune and in Mumbai in the 70s and 80s. But since then, he has worked on mechanisms involved in the development and functioning of the immune system, initially in India at Haffkine Institute, and then overseas at the London School of Hygiene and Tropical Medicine, then at Brandeis University in Waltham, and at Yale University School of Medicine in USA. And then he has worked as a faculty member at the National Institute of Immunology in New Delhi. Currently, since his retirement, he has worked in an honorary capacity at the Indian Institute of Science, Education and Research, that is IISER Pune, at the Translational Health Science Technology Institute in Delhi, at the Christian Medical College in Vellore, and at Ashoka University in Sonipat. His work broadly focuses on the physiological control of the generation, activation, and function of the lymphoid and myeloid cells of the mammalian immune system. He also works with the government on issues of science and society-related policy, with small companies on drug discovery, and with civil society groups, especially those involved in science education and those working more broadly at the interface of science and society. Sir, we are truly honored to have you here today on this online platform. Just before I hand it over, a few quick reminders to all the participants. I request all the participants to kindly mute your audios and turn off your videos for the time being. And later, if you wish to speak, we will call you or invite you, and then you may kindly unmute and speak. And since this is a Q&A session in nature, so please post your questions in the chat box. And with that, now without further ado, since we are all looking forward to start this session, I request Dr. Satyajit Rath to kindly say a few words before we start the Q&A session. So over to you, sir. Thank you.
Thank you very much to Yasmin and to all the other colleagues at Mumbai College. I'm sure I'm going to enjoy myself. I don't know about everybody else. As Yasmin pointed out, I would very much rather that this was a conversation rather than a lecture. So in that sense, I'm going to start out with simply flagging a few points about SARS-CoV-2 and COVID-19, starting with my usual pedantic point that I keep making everywhere, that we should not be referring to this one particular virus as coronavirus or corona. We should be referring to it as SARS-CoV-2 because there are a large number of members of the coronavirus family, and we should be sure whom we are calling names. So in that spirit, SARS-CoV-2 and COVID-19. So apart from nomenclature, let me make a few very broad points. They're not necessarily immunological points, but they are broad points that I hope will contextualize our conversations. Firstly, it is correct that we all across the world are responding to this pandemic with worry and caution. However, there is something that all of us need to understand that we are perhaps, many of us, losing sight of. And that is that this virus infection is not so much of an individual risk as it is a societal or community risk. In explanation of that, let me point out that the likelihood, if you are infected by SARS-CoV-2, of your getting severe infection is certainly less than 20%. I suspect that that's extraordinarily cautious. I suspect that eventually, when all the numbers are in, it will turn out that the overwhelming majority of infections result in asymptomatic or mildly symptomatic consequences. And therefore, the likelihood that you'll be severely ill with SARS-CoV-2 infection individually, leave alone that you'll turn up dead, is very, very low. Unlike, say, other cousins of SARS-CoV-2, such as SARS-CoV-1 or such as MERS-CoV, both of which have a very substantial fraction of likelihood of mortality, meaning that about anything between 10 to 30% of people infected have been estimated to die of a combination number range of SARS-CoV-1 and MERS-CoV. Compared to that, SARS-CoV-2 is eventually going to turn out to be certainly less than 2%. My guess is certainly less than 1%. And under those circumstances, it's not an individual risk for most of us. It's not something that we should be hiding in a corner of the room in terror. We should not be worrying about our own health and safety individually with respect to this virus. What we should be worrying about is our communities. And that we should be worrying about in two different ways. Number one, because we are all naive. This is a new virus, and therefore none of us seem to have any substantial built immunity because of prior exposure. And this is a question that will keep coming up in our conversation. All of us are equally susceptible. And since the ability of this virus to transmit itself between individuals is pretty good, it's going to spread quite rapidly. It does spread quite rapidly. And under those circumstances, even if a small fraction of us becomes ill, all of those numbers will do come up, as we saw in Italy, in Spain, in the US, in New York, in a very short period of time. And that means that the supportive therapy that those severely ill patients need, those hospital facilities tend to be overwhelmed. And then people begin to die needlessly when they should not really be dying. And that's a societal problem for us. That's a community risk for us. And therefore, we need to slow down the rate of transmission so that our medical facilities will not get overwhelmed. Similarly, we need to keep the transmission down because this virus seems to be severely morbid for people with comorbidities, for the elderly, for people with other illnesses such as other lung illnesses, such as hypertension and diabetes, and so on and so on. And therefore, you want to keep them away, people with comorbidities, away from catching the virus as much as you can. But all of this adds up to the virus as a societal risk, not as an individual risk. That's one point. And therefore, this is not something that we should be individually afraid of. This is something we should be cautious of, all of us together. And all of us together matters. Therefore, we should not be ostracizing or blaming people who have the infection. We should be encouraging people to be identified so that appropriate containment can be done, not so that blame can be assigned. The second point I'm going to make before we start is a broad point about what sort of treatments work for viruses and for SARS-CoV-2 and COVID-19 in particular. So broadly, we need to remember that at the moment, there are no drugs, there are no medications that specifically work against SARS-CoV-2 virus. All claims to the contrary, including claims from people in responsible positions making irresponsible statements, such as the President of the United States, hydroxychloroquine is not yet proven to be effective. A couple of drugs, particularly remdesivir, which has been shown in one clinical trial, maybe 1.5, to be somewhat effective. All of that is, in the first place, research in the making, clinical trials that are ongoing. Are we likely to find some drugs that are useful? Yes. Do we have definitively proven drugs which are useful? No, we do not. Any claims to the contrary today, as we speak, are ill-advised claims. The question then arises, people ask me this, how are people getting well? Hey, we have the common cold. In fact, a lot of our common cold is driven by cousins of SARS-CoV-2, other members of the coronavirus family. And we don't need antiviral treatment for the common cold to get well. We get well. So also, we get well with COVID-19. Most of us, even if we have symptoms of COVID-19, will need no treatment and will get fine, will be fine, will recover, will deal with the virus. Some of us, and we can talk about this, some of us will have severe tissue damage reactions, and for that, we will need supportive therapy to keep our organs functioning so that our body and our immune system can deal with the virus and bring us out of it. So what we are really looking for is good, efficient, supportive therapy. And the final point I'm going to make before we start question and answer is very specific about immunity, because everybody is talking about how do we boost our immunity such that we don't get infected. So you don't boost your immunity such that you don't get infected. By and large, your immune response responds to infection coming in, number one. Number two, you really do not want to heighten the level of activation of your immune system. Your immune system is quiescent so long as nothing comes in. When something comes in, you want it to generate a response that is appropriate both in quality and in quantity. Now, all of us have variations in this. Some of us are immunosuppressed, people with transplantation, people with immunosuppressive therapy with autoimmune disease, people who are under cancer chemotherapy, and so on and so forth. But other than such people, none of us is immunocompromised, and none of us needs our immune system to become hyper-responsive. There is a cost to making the immune system hyper-responsive, leave alone making it activated. And that is, the immune system activated and hyper-responsive begins to consume energy and begins to respond inappropriately and cause inflammation. So none of this is necessary. So for everybody, can we all please calm down? Can we all not be terrified, but simply careful and cautious about slowing down the rates of transmission of SARS-CoV-2 and COVID-19 so that as a community in solidarity, we emerge from this safe and stronger together. So I'm going to stop there, and I'm going to leave it open for questions, and then we can talk about questions on the context of all of this.
Good morning, sir.
Good morning.
The first question we have is, could we please have an update on the vaccines in relation to this virus, and what is the reason for its delay?
Right. So, all right. So this is the reason why I did not say anything in my introductory comments about the vaccine, because I was certain that we are going to talk about vaccines. So great. Thank you. So let's take the second part of the question first. What's the reason for the delay? As and when we get a vaccine for SARS-CoV-2, and please note I said as and when, not if and when, because my estimate as an immunologist is we will get vaccines in the plural, and I'll explain all of that. But as and when we get vaccines against SARS-CoV-2, they will be the fastest vaccines ever made in the history of mankind. So this anxiety we have about the delay in making vaccines is somewhat overblown, to be polite about it. We've known about the virus for 6 months. If we have a vaccine in a year from today, that will be a time span of a year and a half between identifying a completely new virus and making a vaccine for it of reasonable efficacy, reasonably broadly available. That, if you think about all the hoops that a vaccine candidate needs to jump through, an extraordinary achievement of mankind. So please don't lose sight of that basic fact. Now, let us think about vaccines. And in order to think about vaccines, let's think about how we deal with viruses and how vaccines help us deal with viruses better. So in order to deal with viruses, we have two components in our immune system that help us deal with viruses. One is the response of our so-called innate immune system. So every cell in the body, pretty much, has an antiviral mechanism that responds to viral infection. Some cells do it better, some cells do it worse, but essentially those responses are dependent on a type 1 interferon response, an antiviral interferon response that uses interferon alpha and interferon beta to go on and prevent viral replication in a variety of ways. That's innate. That our bodies will do even if an entirely new virus comes in. Might do it a little bit better, a little bit worse, but that's what our bodies will do. That's not a response that we can really usefully prime with a vaccine, because that's a generic response to viral infections. So that's the innate immune response. However, the difference between a new virus and a familiar virus is that our adaptive immune system, which generates very individual virus-specific responses, has kind of seen a familiar virus before and therefore will have already made some response that will come useful. This is not the case with something like SARS-CoV-2, which is a new virus. So how do we respond in an adaptive immune component pathway to a new virus like SARS-CoV-2 in a protective fashion? So think about what the virus does. You have a virus particle, you have a cell. The virus particle is inert. There are no active biochemical processes going on in a virus particle. And in that sense, if you like, the virus particle is the non-living part of the life cycle of the virus, although let's not get caught up in the semantics of living and non-living. So this virus particle, in order to go one step further in its life cycle, must come and bind to cells that it's going to infect. So this binding interaction is going to be a specific interaction that a protein on the virus surface will bind to a receptor on the cell surface. When this happens, the virus enters and the virus is going to start taking over the biochemical machinery of the cell. It's going to put viral copies together. is going to make viral DNA, or in this particular coronavirus case, RNA. It's going to make viral proteins. It's going to package virions, and it's going to release them. So one way that the adaptive immune system has of interfering with this viral life cycle is to interfere at the stage of the virus particle binding to the target cell. And the way it will bind to the target cell has to be a way that interferes with the virus particle binding to the target cell. Now, the virus particle is, in molecular terms, pretty large. So you have many potential targets on the surface of the viral particle. Some of these, if you bind with antibodies, it's not going to make any difference to the specific part of the viral surface, the specific protein domain of the virus surface, which binds to the cell surface. So all of those will be antibodies against the virus, but they will not be useful antibodies because they are not going to interfere with the virus binding to the target cell. Antibodies against particular parts of the virus surface will, however, interfere with binding. And when they interfere with binding, you will have viral neutralization because the virus will be prevented from infecting a target cell. And these are called neutralizing antibodies. And by and large, this is a protective category of antibodies. So if either natural infection and or a vaccine can generate protective antibodies directed against a particular part of the viral's particular surface, then you have protection. The second part that we are not going to talk about very much because that's even today not how vaccines are made is, if you have a virus that has avoided whatever else you've managed to do and has gone in and infected a cell. Remember that inside the cell, the virus particle breaks up and the viral RNA, in this case, begins to do whatever it is doing. The virus at that stage has no independent existence inside the cell. So if at that point, the cell itself is broken up, the cell dies and is broken up, then the virus is dead. And it's at that point that the second component of the antiviral adaptive immune response, the so-called cytotoxic T cell response, comes into play. Because if specific T cells can recognize the virally infected cells at a relatively early phase during viral infection and can kill the target cell. So are we killing our own cells? Sure, but we are killing infected cells. And if we kill the infected cell early enough, then there is no virus. And you've therefore eliminated viral infection in a pretty definitive fashion. Despite the fact that this sounds pretty nice, by and large, all vaccine methodologies against viral infections, with the doubtful exception of some HIV vaccine candidates, but certainly all successful antiviral vaccines seem to be dependent on antibody responses, neutralizing antibody responses. So here's how currently SARS-CoV-2 vaccines are being made. They have been, there's, I'm forgetting 140 or 160. I looked at this a few days ago, and there are 100 and something efforts across the world to design vaccines. Pretty much all of them are designed to generate neutralizing antibody responses. So in order to generate a neutralizing antibody response, what do you need to do? You need to introduce the target. Remember the target? The specific part of the virus that's going to bind to the cell surface, and therefore antibodies against that particular part will be successful in neutralizing infection. So what you are going to try and do in a vaccine design is to introduce that part into the body without its being infectious or problematic in terms of causing disease, and to introduce it in such a way that the immune system responds to it and generates antibodies. So you can introduce viral bits into the body in an immunogenic fashion, in an immune response generating fashion, in a variety of ways. Some of these ways are almost 200 years, well, certainly 150 odd years old. They go back to Louis Pasteur. Some of them are only 20 years old. And it's interesting, as biologists, it's interesting that the entire range of technologies for introducing viral material into the body in an immunogenic fashion, that entire range of technologies from 200 years ago to this century are being deployed against SARS-CoV-2. So broadly, there are two categories that you and I can imagine how such viral material can be insinuated into the body, can be introduced into the body. One is to take the viral material, put it in a test tube, put it in a syringe, and inject it into the body. The second is to take the information that codes for this material, the protein by and large, and to introduce that information into the body such that the body will both make the viral material and will respond. So two broad categories of vaccine technologies. First, make the viral material outside and inject it. Second, take the information for making the material and provide the body with the information and allow it to make material and respond to it. You can obviously see that the first category is of older technologies, the second category is of newer technologies. So in the first category, the simplest thing to do is simply to grow the virus, to inactivate the virus or to attenuate the virus so that it's no longer dangerous, and to inject it. So there are examples of this having been proven in monkey experiments from Sinovac, which is a Chinese company, for example. There are a couple of others, but certainly Sinovac has shown this, that beta-propiolactone inactivated SARS-CoV-2 virus generates protective antibody responses, and they've started a clinical trial. A second way of making viral protein outside the body and then introducing it is, rather than taking whole virus, to take the sequence of the virus that codes for the receptor binding protein, to express it in bacteria or in insect viruses or in yeast or whatever, and to make recombinant protein in a biofermenter, purify that protein, put it on adjuvant. Everybody remembers having taken TT shots, and if anybody is being brave enough to actually look at the syringe in the TT shot, the thing going into our arms is opalescent, is sort of turbid. And that's because it's got alum in it, which functions as an adjuvant. So you can take recombinant SARS-CoV-2 protein, put it on an adjuvant, inject that, get a response. This is how many modern vaccines that we are all familiar with are made. And there are efforts out there making such recombinant protein-based vaccines for SARS-CoV-2. And at least one of them, I think, is in monkey trials and is preparing to go into clinical trials. And I'm sure many others exist. So these are the older technologies of making viral material and injecting it. Now, the newer technologies of giving the body the information to make the viral material and respond to it, again, divide into two. One is to put that information on a carrier virus and allow that carrier virus, which is harmless, to infect our tissues very abortively, but enough that viral protein is made and a response is generated. So in order to do this, you can use carrier viruses. Very commonly, adenoviruses or adeno-like viruses are used. And something that everybody will have heard of, which is a chimpanzee adenovirus that is used by the Jenner Institute in Oxford, is currently under clinical trial, having been shown to protect monkeys, although there is some room for concern with the data, but not very much. So they are in clinical trials, and the Serum Institute of India, which is a private company of the Poonawalla family here in Pune, which is one of the world's leading manufacturers of generic vaccines, is in advance scaling up the manufacture of the Oxford vaccine, I gathered from news reports. So that's the third category of SARS-CoV-2 vaccines, and that's the stage at which it is. Rather than providing information through a virus, you can simply take either the DNA or even the mRNA of the viral protein sequence and inject it directly. And people have, over the past 20 years, developed these technologies for a variety of other viruses. And those technologies have been very rapidly deployed for SARS-CoV-2. There are people making DNA vaccines against SARS-CoV-2. There are people making mRNA vaccines against SARS-CoV-2. In fact, another SARS-CoV-2 vaccine that's very much in the news from Moderna, which is a company in the US, is based on mRNA injection directly. All of these are likely to work to some extent. Will they all work beautifully? Will they all protect everybody? Probably not. But a first generation vaccine that provides respectable protection for at least some period of time, we're likely to get information about more than one vaccine candidate by the end of this calendar year, is my considered estimate of this. You will note that none of this involves particularly new conceptual science. These are all technologies that immunologists, vaccine designers, people working in infectious diseases have known about and deployed for a long time. They are simply now being deployed for SARS-CoV-2 in accelerated fashion. And this is what I meant when I said that this is going to be the fastest ever set of vaccines generated. As you can imagine, my long explanation is why I am thinking that we will get more than one vaccine to be successful. We will not get any vaccine that provides 100% protection for the rest of your life, but we are going to get first generation vaccines. They will improve. Here is the real difficulty. The real difficulty is if all 7, 8 billion, whatever of us are there on the planet today, need to be vaccinated. And given the socioeconomic differences between us, if all of us have to be able to afford the vaccine, then the intellectual property rights, the patent related issues that allow private sector pharmaceutical companies to make large profits out of these, what are called blockbuster inventions and interventions, will need to be dealt with, and will need to be dealt with rapidly, and will need to be dealt with in favor of the poor of this world. And that, I suggest to you, is going to take the longest time, much longer than it will take the vaccine technology developers to actually come up with a vaccine. Keep in mind, final point about vaccines, keep in mind that in order to develop a vaccine, typically, classically, what is needed? You need to show in non-human animal studies some reasonable evidence for the protective ability of the vaccine candidate. Then you do human vaccine trials, which means the phase 1 trial, which is of safety, the phase 2 trial, which is of immunogenicity, reliable immunogenicity, meaning that the vaccine is proven to be safe in a few people, then it is given to, and these have to be done in serum. So you show that it's safe, then you show that it's immunogenic in a reasonable number of people, meaning that it generates an immune response reliably. Then you do a phase 3 trial, which in a large number of cases of normal people, you immunize them, and then you look to see whether they are truly protected in real life situation against the viral disease, COVID-19 in this case. And it's only after that that regulatory authorities should and will license the vaccine. And that's the point at which a vaccine candidate will become an actual vaccine. Now, it's possible that in order to get all these data, it might take a long time. And therefore, it's possible that regulatory authorities across the world might actually change their processes such that when safety and immunogenicity and a few other correlates of protection are demonstrated, that they might begin to license the vaccine. This is currently under discussion. There are no rules about this that are completely formulated as yet to make these sorts of exceptions. All I'm pointing out at this stage is just how quickly the vaccine is being made. So this is the scenario of how vaccines are made, what the issues are, and how soon we are likely to get to vaccines. I apologize, I'm answering single questions at great length, but I thought a vaccine question needed a substantial answer.
Hello.
Yeah.
Hello, sir. We've got many questions in our Zoom
I
chat
can't hear
box.
you.
Hello.
Yeah.
So we've got several questions in our Zoom chat box already.
Yeah.
So the first question is by Mr. Siddharth Nakwa. I would want to ask him, Mr. Siddharth, would you like to go ahead and ask the question? You could unmute yourself to do the same.
Yes, ma'am, sure. So my question is, how effective are the homeopathy pills which are provided by the BMC, which are arsenic ALB30 and camphor 1M?
Yeah. So the simple and short answer, I already answered this generically earlier on. Let me say this specifically. Unless something is formally tested for efficacy, we have to assume that it is not efficacious, right?
Yes.
Under that straightforward logic, they're not efficacious. Simply not because I think or don't think that they may or may not work, but because we have no properly examined result and outcome showing that they are efficacious. This is as true of the arsenic ALB30 pills as it is of hydroxychloroquine, as it is of BCG, as it is of leponavir, ritonavir, as it is of any other drug. Every medication that anybody offers us, we have to ask the question, where is the evidence in a formal, well-designed, statistically reliable trial that it works?
Absolutely.
In the absence of such evidence, we should not be taking for granted that anything works. This is how we would respond in any other situation. It's how we should respond in the SARS-CoV-2 COVID-19 situation.
Yes, sir. Thank you so much, sir.
Achha, if you want, I can modulate because I can see the chat box, so I can simply ask people to ask their questions. Is that okay?
Yes, sir, that would be nice.
Okay.
You can go
So
ahead.
the next question comes from Ritika Gaitonde. Go ahead and ask it.
Good morning, sir.
Good morning.
Yes, my question is there a relation between the BCG vaccine and certain immunity to the SARS-CoV-2?
Yeah. So
It was in the news.
yes, it is in the news. It's very much in the news. It's been in the news ever since some mathematical modelers came up with a very tenuous correlation that does not held up. But the reality is, here's what BCG does. Remember that BCG contains mycobacteria.
Yeah.
Mycobacterial material is very poorly cleared. It's very lipid rich and it takes a long time to be cleared. And Yasmin, where's Yasmin? Yasmin knows as much
Yes, I'm
as I do about mycobacterial clearance. So, and all of you have had BCG vaccination, right? everybody's got a little scar on the top of their shoulder.
Yeah.
In fact, I'm probably the only person here who hasn't had BCG vaccination, and I'm too old. There was no BCG vaccination then. So BCG, therefore, not just BCG, any mycobacterium you inject into the body, by and large, causes a fairly large amount of prolonged inflammation. When you cause a large amount of prolonged inflammation, you get a certain amount of bystander immune activation, right? It's actually a cost for the body, because it's not that all of it is needed, but because the mycobacterial material is inflammogenic, you get a fair amount of inflammation. As a correlate of that, it's quite plausible that there are consequences of BCG vaccination, particularly immediately after being injected with mycobacteria, on the ability of the body to respond to other infections, not simply to SARS-CoV-2, but to any other microbial agent. Apart from this, there is no evidence at the moment to believe that BCG vaccination does anything. There are clinical trials, and if the clinical trials show us something different, it will need to be investigated. But at the moment, we have no real reason to believe this. Our current enthusiasm about correlations between BCG and SARS-CoV-2 and COVID-19 is based on an extremely speculative statistical correlation between then rates of COVID-19 infection in various countries and their relationship with BCG vaccination policies in those countries. And that's, for me, way too tenuous and way too transient by way of evidence to be really enthusiastic about.
Okay. Thank you, sir.
Sure. The next question, Sonia Joshi.
Good morning, sir. My question is, has the link between blood group and severity of COVID-19 effects been confirmed, and have any such similar studies been done on the rhesus factor?
So that's a nice question. People have been making noises about this, but I must confess that the last time I looked was a fortnight ago, and I had not come up with any particularly strong evidence in the literature at that time. But I have not looked over the past fortnight at this. On the face of it, it seems unlikely to me to be a particularly strong modulatory factor. But that's simply by way of prior expectations. This is biology, and hey, anything can happen.
Yeah.
That's sort of the Bridavakyam of biology, particularly evolutionary biology. Anything can happen. So might it happen? Well, maybe. Do I expect it to happen? Not really. Do I have up-to-date information about whether literature has tested this or not? I don't. So, so this is one of the questions in which I'm going to say, I don't know whether anything has been reported very recently.
Thank you, sir.
The next question is from Jasmine John.
Good morning, sir.
Good morning.
My question is, how is favipiravir different from remdesivir? Since in my understanding, they both have RDRP as their target.
Okay, so this is not a question that falls within my core domain expertise. So I'm just going to give a very generic answer to this. The absolutely careful domain-specific answer, I wouldn't know. But clearly, the two are completely different structurally. And if my memory serves me correctly, their binding sites on RDRP are somewhat different. So that's why they have been, that's one of the reasons why they've been shown to have a somewhat diverse range of RDRPs of different viruses that they are more or less well capable of inhibiting. So both of them are RDRP inhibitors, but simply that they are different chemical entities and therefore they are differently patentable. You would think that once somebody says find RDRP inhibitors one RDRP inhibitor, another RDRP inhibitor, why on earth should they all get patented? But that's how the pharma company works. But if you want structural information, I don't know the answer to that. The next question is from Riya Nawalkar.
Good morning, sir. I would like to ask you that what do you feel about using mineral solutions?
If you're asking a question, I can't hear you. Okay, so let me just read her question. What do you feel about using miracle mineral solutions in treatment of COVID-19 or to increase immunity? As you can imagine, this is why I said this in the beginning of our conversation. This is what I said in response to the arsenic Calp-30 question. It's what I'm going to keep saying. Unless something has been shown in a carefully conducted investigation to be at least a little bit useful, we have no reason to take on a priori trust the efficacy of any treatment. And that includes the homeopathic remedies that the Ayush department is recommending. It includes the hydroxychloroquine that the President of the United States is recommending. It includes miracle mineral solutions. You know, that's such a nice name, miracle mineral solutions. So no, I do not think that we should take the efficacy or potency of any remedy for COVID-19 at face value. For those students of biology, of life sciences, let me make a sort of academic point about antiviral treatments. Think about it this way. Think about two individuals, both suffering from severe illness, right? But one is suffering from a severe illness as a consequence of a bacterial infection. One is suffering from a severe illness as a consequence of a viral infection. Now, let us assume that I have a beautiful antibiotic that can kill that particular bacteria. And I have an antiviral that can kill that particular virus. And in severely ill, these two individuals, one with a bacterial infection, one with viral infection, I give effective antibacterial antibiotic here and an effective antiviral here. The reality that all clinicians working in infectious disease know is that the antibiotic will lead to dramatic improvement in the clinical state of a severely ill individual. The antiviral will lead to a much more modest improvement. For those of you who have the misfortune to suffer from herpes, from shingles, where you have outbreaks every once in a while of which start with itching and then come out into very painful blisters, sometimes with fever, headache, and so on and so forth. All of these people know they keep both tablets and tubes of acyclovir. Acyclovir works against the herpes virus, works quite well. And yet, all of our unfortunate suffering fellow citizens know that if they do not start acyclovir very, very early during viral infection, if they start it when the first itch arises, it controls infection very well. But if they're a little bit delayed, because they are somewhere and they're not carrying it, and it takes 8, 10, 12 hours. And by that time, if they've had blisters, then starting acyclovir really doesn't work as well. It's not that it doesn't work, but it doesn't work as dramatically. And this is related, I offer you to think about as a speculative point in learning biology. This is related to how bacteria and viruses grow, multiply. Bacteria go, obviously, from 1 to 2 to 4 to 8 to 16 to 32 and so on and so on. But viruses, one virion goes from one virion to what? 1,000? And then from that to another 1,000? In other words, the expansion trajectories of bacteria and viruses are quite different. And perhaps I have yet seen no real evidence that this is why antivirals in advanced clinical morbidity of infections do not have as dramatic an improvement as antibiotics do for advanced morbidity bacterial infections. But I hope it gets you to think about this. What lesson I would suggest we carry from this very speculative part of my argument is for us not to expect too much from any virus-specific medication. Let's all stop looking for miracle cures. Okay. The next question is from Rachita. Do you want to ask your question?
Yeah, good morning, sir. So my question is, angiotensin converting enzyme 2 is present in alveolar cell and small intestine. Now, this is present in higher amount in adults and lower amount in children. And a hypothesis suggests that it has viral receptors. So does this increase the chances of being infected in adults?
Yeah. So ACE2 angiotensin converting enzyme 2 does not have a viral receptor. It is a virus receptor, clearly. If you block ACE2, virus infection is reduced. ACE2 levels are modulated quite extensively. ACE2 is expressed on a variety of epithelial cell lineages in the body at various levels. It's not that children have lower levels. It's that some cell lineages in children have somewhat lower levels. Is this responsible for children having lower chances of infection? It's not even clear to me that children having lower chances of infection is really proven fact for SARS-CoV-2. You see, again and again, we are over-interpreting in our anxieties very preliminary data that change. For example, how do we know which of us is infected and which of us is not with SARS-CoV-2? We don't, especially if more than half of all infections result in asymptomatic infection, then we'll never know. So unless and until we have really reliable modalities by which we can identify people who are infected, we can identify the outcomes of infections in all people, and all of this is going to take a lot of time. A year or so from today, we will have better and better data for this. But at the moment, we should be taking all these claims, the preliminary claim of your question, that children don't have as severe infection or don't have infection, with care. It's not that it is incorrect, it's simply that it's a little unclear whether it's correct or not as yet. Now, is it clear? So what is that children do not suffer as severely from COVID-19 as adults with comorbidities do. So this is not simply a matter of children being protected. This is a matter of children and young adults show no really high frequency occurrence of severe illness in COVID-19. So I'm not sure that ACE2 levels can be necessarily invoked as an explanation for that. Keep something else in mind. ACE2 is not the only cell surface receptor that has been shown for SARS-CoV-2. While it has had the maximum press, there is literature out there that an even more common molecule across the body, cell surface molecule across cells of the body, the SIGIN, I think it's called. It has a numerical nomenclature, it's called CD147, but I think it's called the SIGIN, can also mediate cellular infection by SARS-CoV-2. So whether SARS-CoV-2 works using CD147 and ACE2 together in some cooperative fashion or not is stuff that some friends of mine are working on. And we don't know enough about that. But as a result of the possibility for other receptors for SARS-CoV-2, data with reference to ACE2 levels become even harder to interpret easily. So let's wait and watch as more and more information develops. Next question is from Manasi Prabhu.
Hello, sir.
Yeah, hi.
Good morning. Hello, sir.
Yes.
So my question is, cholesterol is required for the formation of GM1 lipid raft and viruses enter the cell by endocytosis. So, sir, can this lead to slow recovery and increased risk of COVID-19 in patients suffering with cholesterol?
Yeah, so people have been saying this for 2.5 months, right? Ever since March, people have been making this argument about cholesterol-lowering agents, and all sorts of stuff has been said. Again, I haven't tested this in the last week, but until sometime in the previous week that I looked, I have yet to see any really definitive evidence in real life that these correlations exist in the US or in Europe with medication. It's also interesting that you bring this up. There's another point of view from which what you bring up is interesting, which is that viruses that mediate entry through endocytosis would have their endocytotic pathways disrupted by drugs which mess with that pathway. And one of the drugs that messes with that pathway, welcome to it again, is hydroxychloroquine, which modifies the progressive acidification of and early endosomes and late endosomes and so on and so forth. And there is some argument, and there's some in vitro cell culture based evidence to think that that may be a way by which hydroxychloroquine inhibits at relatively poor potency SARS-CoV-2 infection as well as many other viral infections similar. Whether that translates into clinical utility or not is completely an open question at this point. But that is also related to pathways of endocytosis. The next question is from Niharika Gupta.
Good morning, sir. Am I audible?
Yes, you are. Go ahead.
Sir, so my...
I'm sorry, you went off.
To a dangerous immune reaction, which is known as a cytokine storm. So what is the mechanism of this virus and the cytokine storm? Okay,
so let me just repeat the question for people who didn't hear it. Underlying health conditions and chronic illnesses can render individuals susceptible to a dangerous immune reaction known as a cytokine storm. What is the mechanism of virus and cytokine storm? All right, so this is an interesting question because the cytokine storm that everybody talks about so anxiously, as though your immune system is trying to kill you, is actually simply your immune system trying to protect you. So when the virus comes in, we've talked about this earlier in this session, when the virus comes in, we have two kinds of generic immune processes that develop. One is a type 1 interferon response. The other, slightly more specialized, but even so generic, is an inflammatory response. And inflammatory responses are simply the body's way of location containment. You have a virus infection somewhere, you cause an inflammatory response there, and it allows you to sort of seal off the infection, whether it's a viral infection, whether it's a bacterial infection, to seal it off in one part of the body so that it doesn't spread outside. Now, the immune response, the immune inflammatory response that mediates this localization, if it doesn't work too well for a variety of reasons early during infection, the virus spreads, but the body is still trying to contain inflammation, using inflammation, is still trying to contain the virus wherever it finds the virus, or indeed wherever it finds the bacterium. What's going to happen? You're going to begin to have containment, not only in one or two parts of your lungs, for example, but in many parts of your lung. And if it begins to happen has an inflammatory response in many parts of your organs, then two things are going to happen. Enough inflammatory cytokines are going to start getting made cumulatively in all of these locations together, that they are now not going to be restricted simply because of concentrations. They are not now going to be restricted simply to local effects, but they are going to start causing general effects all over the body. And that's one reason why these high levels of cytokines make for severe illness. And a second is, because these cytokines are causing immune inflammations, attempting containment all across your organs, effectively, your organs are beginning to function less and less well. And therefore, you begin to get organ dysfunction or organ functional failure of organs. So the so-called cytokine storm is really the body trying to mediate infectious microbe containment. That it is happening at a stage of the illness when the virus or the infecting microbe has already spread quite widely across at least one organ, in the case of SARS-CoV-2, the lungs, is what leads to severe illness. So the cytokine storm situation is really not an indication that everything is going downhill. It is an indication that the cure is proving to be worse than the disease. And this is why, if some specific components of the cytokine storm, such as, for example, interleukin 6, which is a major member of the cytokines causing a cytokine storm, if we give neutralizing antibodies against interleukin 6 to people suffering from cytokine storms, then the body-wide effects of these cytokines are sufficiently suppressed to allow the body to get on with having the productive immune interaction with the virus that will eventually lead to viral clearance and recovery. So this is the trajectory of infectious agents bringing up cytokines, and at a certain stage of progressive infection, entering into the status of the cytokine storm. So we could talk about cytokine storms a little more, but it would be unfair to other people asking questions. So let me go on. Aishwarya Lonare.
Oh, yes. Good afternoon, sir.
Good afternoon.
So my question is, how can we make use of the antiviral property of copper in order to control SARS-CoV-2 infection?
So the short answer is, at this point, I don't see how we can. Because the antiviral properties of copper depend on copper as a chemically reactive moiety, and copper as a chemically reactive moiety to introduce into the body is going to cause all sorts of major dysfunctionalities, and therefore I can't see any way of introducing copper into the body. You can think of all sorts of ways in which copper surfaces, for example, have been claimed to be relatively more antiseptic in a variety of ways, because copper is a reactive entity. Very many people are working on how to disinfect surfaces in the context of SARS-CoV-2. But we already know pretty good disinfecting agents. Since it's an enveloped virus with a lipid envelope, soap and water work pretty well. Many other reactive agents such as bleach work very well. Just as soap works, chlorhexidine will work quite well since it will disrupt lipid membranes. We already know how to deal with any SARS-CoV-2 that we may have on our hands. Wash your hands with soap and water. So I'm not sure that I can think of really particularly interestingly innovative ways in which to use copper. Avni Nevrekar. Yes, no? Okay, let me ask a question and answer it. The SARS-CoV-2 has been circulating only for 5 or 6 months, which means there's simply no way to know whether immunity to the disease lasts longer than that. How long does it last? Great question, particularly because there are other coronaviruses, but there is some evidence that antibody responses are not long-lived. Let's not talk about immunity lasts or doesn't last. let's talk about what particular part of the immune response is being tested. So the particular part of the immune response in many of these viral infections that's been tested is antibody levels. And antibody levels in a variety of different infections have been shown to last wildly different periods of time. So measles virus antibody levels are known to last for very, 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 very long periods of time, years and years and years. But there are other viral infections, such as, in fact, some of the coronaviruses causing the common cold, which don't last very, very long. We have no idea. Honestly, idiot immunologists have still not figured out what the difference is that allows one to last for longer than other. We have all sorts of, I will not call them, I will not dignify them with the title speculation, hypothesis. I'll just call them speculations. Immunologists have speculations about why there may be differences, but I have yet to see a single instance where immunologists like me have demonstrated the mechanistic basis of why antibody responses to one virus last for a very short period of time and antibody responses to another last for a long period of time. In terms of evidence, you're absolutely correct. As you point out, we've known about SARS-CoV-2 only for 6 months or so, and we at the moment have no idea on how long antibody responses are going to last. As a final modification, not only do we not know this, we don't even know whether everybody's antibody responses are going to be as potently neutralizing antibody responses. Remember what I said at the beginning? Virus particle, you can make antibodies against all sorts of parts of the virus particle. But unless you make antibodies that interfere with the virus binding to a cell to initiate its life cycle, the antibody response may not do a great deal. It might work a little bit, but it may not be particularly potently protective. So we have no idea of protective potency of antibodies and how variable protective potency is in the antibody responses, leave alone the idea of about how long the antibody responses last. So all of this is science in the making. The next question is Manasi.
Satyajit,
Yeah.
Satyajit, can I interrupt here? Can we take a couple of questions from the list that came to us earlier?
Sure.
And then we'll continue with this.
Sure. Go ahead.
Good afternoon, sir.
Good afternoon.
Okay, so the question is, when COVID virus enters through mouth, it has two destinations, intestines and of course lungs. But why diarrhea is not so common symptom for coronavirus disease? And why is it preferring lungs epithelial cells, although enterocytes of the small intestine also have ACE2 receptors?
Yeah.
Also, can it enter through damaged skin cells? Would Langerhans cells could destroy it?
So the short answer is that the life of the virus is bound in transmission terms on air droplets exchanging between people. And air droplets don't go down our esophagus anywhere near as easily as they go down our respiratory pathways. And my guess is, I don't know this for a fact, but my guess is that the explanation for why it infects the way it infects is going to be as mundane as that. Because it gets there, it sets up an infection. I don't think that it is going to be particularly easy for an enveloped virus like SARS-CoV-2 to resist gastric acid. And therefore, I suspect that intestinal infection is not going to be common. Although I hasten to add that one of the symptoms for SARS-CoV-2 infection increasingly in the sort of minor but noted list of symptoms is indeed diarrhea. And the argument has been made that that may be mediated by the fact that, as you point out, ACE2 molecules are present on a wide variety of epithelial cell surfaces. But I don't think that there's going to be much more to it that's respiratory cell lineage specific than simply the mode of transmission. A lot of biology is as much supraorganismal pathways as it is cellular and molecular pathways.
Hello, sir.
Hi.
The next question we have is, why is the R naught for SARS-CoV-2 much higher than the other viruses that have caused epidemics or pandemics?
So it's actually not anywhere near as high as many other examples. Measles, for example, has an incredibly high R naught. For people who don't know what R0 is, it's the rate of transmission in very crude terms, which is all I can manage since I don't know either statistics or mathematics well enough to understand epidemiological modeling well. But for those of us who are in my boat, let me put it in terms that you and I can understand, which is the R0 is the measure of how many individuals can one infected individual transmit infection to. And obviously, once somebody tells us that number, let's not worry about how they calculate it. Once somebody tells us that number, we can imagine that if the R0 is less than 1, then you have an infection in a declining trajectory in the community, because one cohort of people is going to infect a cohort that's smaller than them, and then that cohort is going to infect a group, a number that's smaller than them, and the infection will die out in the community. Which is why how large R0 is grounds for great concern, particularly for theoretical epidemiologists with respect to this. The R0 calculations, I suspect, are somewhat less precise than theoreticians would have us believe. But that said, because the R0 is a real life calculation, so it depends on whether pollution will facilitate or inhibit transmission. It depends on what the age structure and distribution of the population is, so that it depends on how long a given individual remains infectious. It depends on the socioeconomics of, it depends on the socioeconomics of Dharavi versus Malabar Hill. So in all of these ways, the R0 is a real life calculation. Biologists, cell and molecular biologists, have this overweening arrogance to claim that they can explain through purely cellular and molecular pathways why two different R0s are different. And I caution you that their arrogance may not always be justified. That said, that caveat made, a simple argument is that the better a virus is at resisting obvious environmental inactivation, such as through free radicals, through temperature shifts, through drying on the one hand, and the higher the affinity of viral surface receptors is for the cell surface targets, the more efficient infection is going to be. Therefore, the more efficient transmission is going to be. Therefore, the higher R0 is going to be. And for SARS-CoV-2, particularly, this argument has been made that the spike protein of SARS-CoV-2, the receptor binding domain of the spike protein of SARS-CoV-2, the receptor binding domain has now become a foolishly acronymous word by itself. Everybody refers to RBD. So the RBD of the S protein, which is simply the receptor binding domain of the spike protein, is responsible for binding to ACE2, and that affinity is pretty high, especially as compared to other coronavirus spike proteins. And that's been argued to be one explanation for this. So let me stop there because I tend to go on and on. Go ahead.
Hello?
Yeah, go ahead. Are you going to ask me questions, or shall I go back to the chat box?
No, so there was one question to be answered.
Go ahead.
It said that there was a case in Gujarat in which a woman gave birth to two twins, of which one child tested positive and the other tested negative. How was that possible?
Why not? In a household, when you, I mean, I go out, I get infected, I bring it back. There are 5 people in the household. with all of whom I'm sharing two rooms in the household. Yet all 5 don't necessarily get productively infected. Let me point out that in all of these situations, not 2 twins, 1 twins. In all of these situations, 2 twins is that. In all of these situations, what we are detecting is virus presence. For all you and I know, both babies got infected. One dealt a little bit more rapidly with the virus compared to the other, so that at the time of testing, one had already cleared the virus and the other was on the verge of clearing the virus. So this sort of, you see, we are trying to, sorry, I've had voltage fluctuations. We are trying to provide an explanation by mechanism for every statistical phenomenon. But the whole point of statistical phenomena is that they are probabilistic, that each individual data point cannot be definitively explained. So keep that in mind when we struggle with trying to explain these kinds of situations. It's not so much that I cannot provide any explanation, it's much more that I can provide far too many possible explanations for it to be useful in understanding. And this is our limitation as biologists and academics that we should acknowledge and keep in mind. Go ahead.
Thank you, sir.
Okay.
This will be the last question from the list, after which we will get back to questions from the chat box.
Okay.
The question goes, how different is the immunological response of the body to COVID-19 as compared to SARS-CoV and MERS?
Aha. That's a great question. It's a question that very few people in the world are in a position to answer. One of those is at the Erasmus University in the Netherlands, a gentleman named Bart Haagmans. How does one even begin to answer this question? So there are 2 ways of beginning to answer this question. One is to take all the old information that has been collected about SARS and MERS patients, of whom there were not very many, there were a few thousand. The technologies for investigating immune responses at that time were quite different from the technologies that are available today. So it's in those ways that there seems to be a difference in even beginning to get at this question. Clearly, SARS and MERS caused very severe illness more easily than COVID-19 does. Remember, I'm referring to the diseases. SARS is the disease, MERS is the disease, COVID-19 is the disease. SARS-CoV-1 is the virus, MERS-CoV or MERS-CoV-1 is the virus, and SARS-CoV-2 is the virus. I know people who are attempting to correlate immune parameters that were collected at that time versus those that are being collected now. I have yet to see any evidence, any analysis that provide insights on why SARS and MERS were far more severe illnesses than COVID-19, but SARS-CoV-1 and MERS-CoV-1 were nowhere near as easily transmissible as SARS-CoV-2 seems to be. At the moment, all we have is these speculations and no real answer. What Bart Haagmans has been doing about this is interesting. So 20 years ago, 17, 18 years ago, 2002, 2003 was when we met SARS-CoV-1 for the first time. Ever since then, the Haagmans group has set up a non-human primate model of SARS-CoV-1 infection in cynomolgus macaques. And cynomolgus macaques infected with SARS-CoV-1 have been analyzed quite extensively in the Haagmans lab. The Hagman's lab is now infecting over these past 3 months cynomolgus macaques with SARS-CoV-2 and are just beginning to do compare and contrast analysis. Again, at the moment, we have no really interesting information coming out of it, with one exception that I'll come to in a moment. But this is how we learn about mechanisms. We don't learn overnight. We don't learn with an easy, obvious, self-evident trajectory. We bump our way through to understanding mechanisms. With all of this, so the short answer to the original question is we don't know, as is evident to everybody. So, but let me give you one example that I am essentially putting together from the Hagman's papers. So I'm dealing with papers 10 years apart, almost. I'm not sure exactly 10 years, but about 10 years apart. The recent paper, which is from 15 days ago, from Hagman's, essentially demonstrates that cynomolgus macaques can be infected with SARS-CoV-2, and while they don't seem to be clinically ill, do show lung lesions that are kind of sort of similar to those observed in rhesus macaques in experiments in the US and those observed by the Hagman's lab in SARS-CoV-1 infected cynomolgus macaques. All very good. Now, think about 10 years ago when the Hagman's group published a remarkably interesting paper on SARS-CoV-1, mind you, not SARS-CoV-2. No, there was no SARS-CoV-2. When they infect with cynomolgus macaques with SARS-CoV-1, the monkeys are not clinically ill. However, there are some monkeys that are clinically ill, a little more severely ill. And the more severe illness in cynomolgus macaques correlated with the age of the macaque. And that reminds you of, hey, SARS-CoV-2. Age is supposed to be a significant comorbidity for COVID-19. And now, there is a very interesting correlation in the old paper, mind you, that not just me, everybody is looking back at, which is that the cytokine response of older macaques to early infection is a little bit less type 1 interferon, a little bit more generic localizing inflammation. Is this significant at all for SARS-CoV-2? Is it one potential factor explaining the role of comorbidities in severe illness in COVID-19? We don't know. But those are the kinds of things that I suspect the field is going to be focusing on, is already focusing on, and is going to be focusing on more and more as we go along. Okay, let me go to the written questions. Manasi Prabhu.
Hello, sir.
Hi.
Hello.
Yeah, we can hear you.
Sir, remdesivir and some molecules available for treatment of HIV are in the process of being combined for checking activity against coronavirus. So, sir, do you think that, this combination will be able to match similar specificity as the antibodies produced by the rare B cells of COVID-19 patients?
So remember that antibodies work differently. Remember what I said. Virus is binding to cell, the antibody is going to prevent the virus binding to cell and setting up infection. All the drugs we are talking about, drugs that are directed against viral proteases that processes the polypeptide that the virus makes into foldable and usable proteins, drugs that work to inhibit the RNA-dependent RNA polymerase, which is what allows the virus to expand. All of that is happening inside the cell after this step. So because antibodies and these drugs work in different locations, we really can't be comparing the potency and so on and so forth of these steroids. That said, let me reiterate that none of the antiviral drugs that have been repurposed from HIV treatment for testing in SARS-CoV-2 infections has formally been demonstrated to be particularly useful. Most of them are still under clinical trials. I'm aware of 1 or 2 clinical small trials that have shown some signs that they may be useful. But a general lesson from all the clinical trials that we've seen so far for anti-SARS-CoV-2 drugs is that even when we will find, and I'm sure we will find, that some of them work, the effect size when they work is always going to be modest. We are not going to find a miracle cure. We are going to find combinations that help. Let me quote, since Anthony Fauci brought up remdesivir in one of his press briefings, rather than through the formal academic pathways of reportage. Let me point out to the remdesivir clinical trial that he was referring to. Improvement did not mean people got well or people got well immediately. What improvement meant with remdesivir in the successful clinical trial was the median time for leaving hospital went down to 11 or 12 days from 14 or 15 days. The mortality went down from 11 or 12% to 7.5 or 8%. These are not dramatic effects in terms of the size of the effect. Keep that in mind when you look at hyperbolic newspaper reportage about, oh, here's a new, potentially, what was Mr. Trump's word? Game changer treatment. My guess is that game changer is going to be up to us to be sensible, to keep physical distancing, not to keep social distancing. India should not use the phrase social distancing, given that for 2 millennia, we have used the caste system as a means of social distancing. We should not be using the phrase social distancing. We should be using the phrase social solidarity, but with physical distancing, with a simple barrier to exchanging droplets, and being careful, being aware, being cautious with these, staying socially inclusive and together, and pressing our governments to ensure that appropriate public health expenditure is done, that appropriate drugs and vaccines are made available to everybody. It's that kind of citizenship that's going to be the game changer. Drugs are not going to be the game changers. Vaccines will, but drugs are not going to be the game changers. Okay, the next question is from Jasmine John. Didn't you already ask a question?
Yes, sir.
Okay, go ahead.
We can go ahead, I think, with the next one. We should give everybody a chance.
Yeah, which was what I was implying. Duria?
Yes, sir.
Go ahead.
So my question is, interferon alpha 2b has been used to treat diseases like HIV and dengue and hepatitis in the past. So could this be used as an effective treatment for SARS-CoV-2? I'm sure, sir, you've been answering the similar questions for long.
No. Great question. So thank you. So let me add a caveat to my claim that no treatment is going to be very large. The caveat is, I stay by that statement. But if you look at within the modest effects of therapies, the kind of sort of best one comes from this interferon, this type 1 interferon therapy. So you're absolutely right. Interferon has been argued and there's one clinical trial out there that says interferon works. It's a convoluted clinical trial because it uses anti-HIV drug treatment and then adds interferon on top of that. So there are all sorts of difficulties with that trial, but the fact remains that there is some evidence out there to think that interferon therapy might work. Now couple that with the Bart Hagman's cynomolgus macaque data I told you about from 10 years ago. Repeat, they are about SARS-CoV-1, not about SARS-CoV-2, so they may not apply at all. But the fact is that comorbidities may in fact be associated with a somewhat of a quantitatively lesser type 1 interferon response. And combining all of these together, what you're saying is very much what the field is working on, to the point that type 1 interferon therapy is being clinically tested. In one trial, it has been reported. I'm aware myself of at least 5 other trials where it is being tested in a variety of combinations. And I suspect that along with many of these other antiviral drugs, type 1 interferon therapy will also show modest but significant effects. And in combination, as weeks and months go by, we will have better and better clinical interventions to offer. None of them is going to be by itself a huge game changer, but together they will make differences that are hugely meaningful to individual lives at stake. The next question is from Niharika Gupta. Didn't you ask? No. I'm now getting confused with names. Go ahead. If you didn't ask, go ahead and ask.
Good afternoon, sir.
Hi.
Sir, so my question is, what are the roles of immune cells from the adaptive T cells and the innate system, such as the natural killer cells and the T?
Okay, so I kind of answered this question when I responded to the vaccine question way back. So let me just recapitulate. You have, when viruses come in, you have innate immune responses. One of the early cellular innate immune responses that you're going to get is a natural killer cell response that will kill viruses. For SARS-CoV-2, in fact, for coronaviruses, we don't know enough about coronavirus immunity in terms of the role of NK cells played in them. My guess is, like very many other viral infections, NK cells play a modulatory, small but significant role rather than a huge role. But we have no real information about that. The innate immune system I already talked about, type 1 interferon responses, preventing cellular viral infections, and inflammatory responses, classical inflammatory responses leading to localization of infections. The T cell responses I talked about as cytotoxic T cells, identifying infected cells and killing them and eradicating virus. And the antibody response, which is mediated by both CD4 T cells, which will help the B cells make a high affinity potent antibody response, together preventing viral particles from infecting new cells. So I'm going to leave it at that because we've already been online for an hour and a half.
Thank you,
while
sir.
I have no problems with going on, that's not necessarily the case for all of you. So please do vote with your feet as and when you would like. And when we fall below a certain threshold or whenever the organizers want to stop, we can stop. Until then,
I was going to ask you, Satyajit, looks like there are a lot of questions. If you have no problem, I think we'll continue for a while.
Sure, I know, as I already said.
Thank you. Thank you.
I'm happy to go for more time. No worries. So Sonia Joshi.
Good afternoon, sir. My question is it safe to use corticosteroid nasal sprays to control the nasal allergies or inhale corticosteroids to control asthma symptoms during the COVID pandemic?
That's an excellent and practical question. And my guess, this is not definitive information that I'm providing. Simply, biologically, my guess is that the immunosuppressive properties of the steroids in preventing allergic degranulation are sufficiently transient that the sprays will probably not make a major difference to SARS-CoV-2 infection in the upper respiratory tract. But you will, I hope, hear my tentativeness in answering this question. Part of it is that I've already asked friends across the world, some of who are working on these sorts of SARS-CoV-2 immunity questions this question, none of us has a really strongly based answer. All of us will say this very tentative thing. So the answer is probably yes, but I'm not completely sure. I would, on the one hand, I would argue that the risks of SARS-CoV-2 exposure in an already inflamed upper respiratory tract, which is what happens with people with respiratory allergies, might in fact counterbalance the effects of nasal steroids. Add to this the little factlet that nasal steroids don't really increase the risk of many other upper respiratory infections hugely. That's been tested. And the effects are really very small. And so I'm using that to make the argument that if at all there is an effect, it might be a small effect. But please hear both my answer and the tentativeness with which I'm giving it.
Thank you, sir.
That was a great question. Avani Nevrekar, hey, you already asked a question. But the answer to the various facilities provided in the hospital is, as you would imagine, supportive therapy, get the lungs to work, get the kidneys to work, and or replace their work with oxygen ventilators, dialysis, whatever. And spread in hospitals is exactly the same way as any other infectious agent. Containment, barriers, containment and barriers. I should also answer Jasmine John's question about surfactant replacement, inhaled nitric oxide, high frequency oscillatory ventilation for newborn treatment. And all of these will have different roles to play. And these are clinical judgments that I really don't know enough about in terms of clinical respiratory medicine to be able to answer as in any really. So the short answer to that question is I don't know. Srishti Vishwakarma.
Good afternoon, sir.
Hi.
So my question is that a lot of research has said that bats and pangolins are intermediate hosts. So do domesticated cats also act as intermediate hosts, and can they transfer the disease to humans?
Right. So, so the question is, do domesticated cats act as intermediate hosts for SARS-CoV-2? There is no current evidence that suggests that they act as intermediate hosts for SARS-CoV-2. That's a formal, technical answer. There is no such evidence available so far that cats can act as intermediate hosts for SARS-CoV-2, that they have done so in the past, or that cats can transmit infection to humans. Now, for neither of these do we have any evidence. However, it is true that the Felidae, members of the cat family, not just domesticated cats, but other members of cat family, including poor tigers in zoos, can and do get infected with SARS-CoV-2. My understanding from this is that symptoms, if at all, are quite modest by and large in cats. And I have been asking, and I have so far not seen or heard of any evidence that has shown cat to human transmission. So we've been infecting cats, but so far there's no, so there is evidence that we've been infecting cats, but so far there's no evidence that cats have been infecting us back. And in that formal sense, they're not intermediate hosts. Ashwai Dantra.
Good afternoon, sir.
Good afternoon.
Sir, my question is, a later vaccine study in mice found that memory T cells protected the animals from the worst effects when scientists tried infecting them again with SARS. Why does this not occur in terms of COVID-19?
We have no idea whether it occurs or not. That's what I meant when I was talking about vaccines. I said that there are T cell responses, and you would imagine that vaccine makers would focus on T cell responses as well. And yet the reality is all vaccine efforts. In fact, most efforts at understanding immunity to viral infectious diseases for a variety of reasons is focused on antibody responses. Part of this is neutralizing antibody responses do seem to work to protect us. Part of it is antibody responses are relatively easier to analyze. So it's a little bit like, you know, where do you look for something you've lost? You look for it under the light pole. It's not quite as bad as that, but there is a little bit of, oh, we look at the antibodies because we can look at antibodies in the human situation quite easily. Looking at CD8 T cell responses in humans is nowhere near as simple and straightforward as all that. So I'll give you an example. Yellow fever vaccine. The yellow fever vaccine is a vaccine that gives us 10-year protections. It's a viral vaccine. It gives us 10-year protections, and people have worked on antibody responses forever and ever. And it's only during the past 10, 15 years that people have begun examining cytotoxic T cell responses generated by yellow fever vaccine. But we still don't have any real life idea of whether yellow fever protection is mediated and to what extent by cytotoxic CD8 T cells. A footnote to this is that keep in mind that most people don't want an infectious vaccine. There are exceptions to that, but by and large, people tend to worry about infectious vaccines to the point that the live poliovirus vaccine falls out of usage in Europe and North America, where injectable vaccines begin to be available if needed. And all of this is because we worry that in an occasional recipient, the weakened but live viral vaccine might actually cause disease. This on one side. On the other side is the reality that CD8 T cells in particular, cytotoxic T cells in particular, are by and large triggered by actively replicating viral infections and not by killed virus material or by recombinant viral protein. And that's in part why vaccine makers tend to focus on antibodies and not focus on CD8 T cells. So the short answer to your question about what about the role of cytotoxic T cells in COVID-19 is we don't know. I've seen only some preliminary data that says that CD8 T cells are in fact primed and triggered in COVID-19 patients. But what their role and significance is, at the moment, we have no real idea. But this is sort of the background to that context.
Thank you, sir.
Thanks. The next question is from Rachita.
Yes, sir. Sir, my question is, delirium has been observed in many patients. So it with other psychological issues can prolong and complicate the recovery. What are your views on this?
Okay, so quite frankly, this is again a deeply clinical question, and I don't have definitive and or authoritative views to offer on this. I'm just going to make a couple of comments about it, but keep in mind that I do not have authoritative and or definitive views about this. Number one, there are speculative claims out there that SARS-CoV-2 can infect cells of the nervous system. I have not yet seen definitive evidence, but I have heard claims to this effect. Whether that's involved in some of this, I don't know. The second is not simply medication. The cytokine storm itself can cause neurological consequences, and that are well known. And there are considerations of that kind that are included in the intensive care therapy protocols that such patients undergo. Can these complicate and prolong recovery? Absolutely. But can I make any more informed comments about this? So I'm going to move on. Krishnendu Nair.
Good afternoon, sir. What is herd immunity and how effective is it against COVID-19, and how does it work?
So I was wondering when something like this was going to come up. So here's the issue. I live in a household of 10 people, say. I go out of town, I pick up an infection, I come back sniffling and coughing and so on and so forth. COVID-19 patients don't really sniffle, so let's just stay with coughing. So I come back coughing, and I come home, and there are 10 people, some elderly, some young, some my age, whatever. And I am coughing at everybody, right? That's our normal style. We cough at everybody, and on the streets, we're spitting everywhere, etc., etc. So I do all of this, and potentially, because it's a new virus that I have met when I went for some meeting in Wuhan, or in New York, or in Rome, I brought back a virus that none of these 10 family members have seen. Potentially, therefore, I can transmit infection to all 10. And potentially, when they go out, when the kids go out to play, when the elders go to the garden, they can distribute them to another 10 people and another 10 people. This rate of transmission is going to shift if, when I come home with an infection, there are 10 people in the house, but 6 of them have already met the same virus somewhere else and have, we assume, become immune and protected. In which case, the virus can only spread from me, not now to 10 people, but to 4 people. So basically, as the frequency of people already exposed in the community grows, as the frequency of people who have been exposed to the virus and are presumably immune to the virus grows in the community, that growth of percentage has a major negative effect on the transmission efficiency of the virus. The virus is transmitted less and less efficiently. So remember what I said about R0. As a result, as the proportion of exposed and immune people in the community rises, R0 declines. And as R0 declines, beyond a point, that increase in proportion of exposed people will bring R0 down below 1, and therefore will lead to a self-limiting trajectory of the viral infection. This frequency of exposed and therefore protected people is the notion of herd immunity. This is how immunity in the herd, so none of us follows herd immunity easily because we don't think of ourselves as a herd. Sometimes we think of ourselves as a mob, but we never think of ourselves as a herd of buffaloes crossing the road at Churchgate Station. So think about it, rather than thinking about it as herd immunity, think about it as community immunity. How prevalent is immunity in the community is the question that the notion of herd immunity is asking. And the more prevalent immunity is in the community, the more likely R0 is to be lower and lower. Transmission efficiency is to be lower and lower. And that's how it works. So in a sense, if you think about it, what all of us are waiting for is for a vaccine to come in, for many of us to get vaccinated, and therefore to become immune. So effectively, the vaccine is a way we have of engineering herd immunity or community. What percentage of people being exposed and immune is the breakpoint for herd immunity to work is a question that really shouldn't be asked that way. Because as you can see in my description, for different viruses with different R0s, different modalities, different potency of immune response, and so on and so forth, these numbers are going to be variable for different infections. Do we know this number for SARS-CoV-2? Not really. So people out there who are saying, oh, 60% will work, oh, 50% will work, oh, it will need 70%. This is all very speculative. But this is the broad qualitative idea of herd immunity. Melanie Shah? Okay. The question is, how does SARS-CoV-2 increase the propensity to develop Guillain-Barré syndrome? So the Guillain-Barré syndrome is thought of as a sort of bystander autoimmune response, and we are at 12:50. So for me to get into explaining neuroimmunology would be way too much. So let me not get into that. I will let you pester Yasmin about explaining neuroimmunology to you from her earlier incarnations. And I will simply point out that any systemic inflammatory process runs the risk of odd little outcomes, such as a neuroinflammatory outcome like the Guillain-Barré syndrome. And I'm really just going to leave it at that. Michelle Pereira.
Hello.
Hi.
Yes, sir. My doubt is more like my question is more like a common doubt that most people might have. Since we've been told that virus stays on surfaces for 2 to 3 days, how risky is online shopping? How should parcels be sanitized, and what personal care could we take in dealing with these parcels? Thank you.
Yeah. Okay. So again, great question. Split that question into two parts. The first part of the question is, we don't care how long the virus stays where. What we really care about, you and I, is indirect transmission. So let me get terminology straight. Direct transmission is, I am sick, I cough, and the droplets go into somebody else's airways. That's direct transmission. Indirect transmission is, I cough on the surface, virus, the droplets dry there, the virus is still sort of kind of there, then somebody else comes and touches the surface, a little bit of the virus rubs off on their fingers, then some more time, indeterminate time later, they touch their nose, mouth, whatever, and some very small part of the virus goes in. This is indirect transmission for the purpose of this conversation. So the question that we should begin with is there evidence what proportion of COVID-19 infections are caused by direct transmission and what proportion are caused by indirect transmission? So let me quote for you. Rather than quoting formal scientific literature, let me quote one of the better pieces of popular science that's accessible. So in an issue of the New Yorker magazine a couple of weeks ago, Atul Gawande, who is of Marathi origin and extraction, but who was born and brought up in the US and is an outstanding public health researcher and a brilliant writer about public health matters, who's written a book called Better, which is very interesting reading in addition to many other books, wrote an article in the New Yorker about SARS-CoV-2 transmission in Dr. Gawande makes a very interesting point. He points out that current estimates are that only about 6% of SARS-CoV-2 transmission is via indirect routes. The overwhelming majority of SARS-CoV-2 transmission infections seem to be through the direct route. So when we begin talking about virus-contaminated surfaces, which everybody is terrified about, we are already talking about, in practical terms, a relatively minor pathway of infection. Couple that with the fact that in many of these instances where people have shown that there is virus survival on surfaces for 8 hours, 12 hours, 24 hours, 48 hours, oh my goodness, 48 hours, and so on and so forth. We're not taking two factors, quantitative factors, into consideration when we interpret those data in terms of what we should do in real life. The first is that virus is detectable is not to say that the same amount of virus is detectable. The amount of virus is steadily going down, even in those studies. And since infection risk is always a matter of dosage, these data don't all indicate an equal and high risk of infection transmission. This is compounded by the second problem, which is that many of the studies of viral persistence on surfaces are not looking at infectious virus. They are looking at real-time RT-PCR detectable viral RNA. But that viral RNA can come from viral particles that have already been damaged and that are therefore no longer infectious. So as a consequence, I am inclined to say to all of us, me included, that we should calm down about this, that we shouldn't get worked up about what's a relatively small pathway of infection and that declines further and further in its significance as time goes by hour. Yet, it is also true that we should worry about infecting people with comorbidities who stand some significant chance of becoming severely ill. But for all of us, simply falling into the habit of washing our hands regularly, soap and water washing our hands regularly, seems to me to be affordable and sufficient in ways in which sanitize, use and throw wipers for every package that we bring into the household from outside may not. So we need to worry about trade-offs, and I would argue that this is how we might want for each one of us separately in our own lives to make judgments about which trade-off would be reasonable. Kshitija Rahate?
Satyajit, we are close to 1 p.m. What I would suggest is, would you like to just glance through those questions and maybe take a couple of them which you think may be very relevant, and then we'll skip the rest.
Okay, give me 1 minute and let me sort of scroll through questions.
Yes, that would be nice.
Question about plasma therapy, we will address. Gender differences, we will address. Chimps and bonobos are not used because they're not available. T-cell responses that we've already talked about. Cross-reactive antibodies we will talk about when we talk about plasma therapy. All right, so let me talk about cross-reactive antibodies and plasma therapy. As I have pointed out repeatedly, all antibodies are not going to be equally protective. The antibodies that are going to be most protective are going to be antibodies directed against the receptor binding part of the viral protein. The receptor binding part of SARS-CoV-2 spike protein shows a peculiar variation in sequence, a 3 or 6 amino acid variation in sequence. That's something similar has been found in pangolins, which is why pangolins have been thought of as one intermediate host. But those are not found in any of our previously exposed 6 coronavirus members. 4 that cause the common cold, SARS-CoV-1 and MERS-CoV-1. And therefore, antibodies that are most protective, which are going to be directed at that part of the spike protein RBD, are not going to be found in response to other coronaviruses, and indeed are not in the studies that have been reported. People have been making monoclonal antibodies for therapeutic purposes, and in all of those, the data seem to indicate that cross-reacting coronavirus antibodies, which can be detected against other viral proteins and other parts of the spike protein, do not really provide particularly efficient cross-reactive protective potency. This brings me to the plasma therapy issue, and in plasma therapy, keep in mind what I said. Each one of us responds quantitatively and qualitatively differently to infection. So how much of an antibody response we make and what part of that antibody response is truly high potency in protection is variable. And because it is variable, a major problem with plasma therapy is how much of my plasma will need to be given to mediate protection in the recipient needs to be tested. We need to come up with tests that are validated before we can think about plasma therapy as an actual treatment. So please do not misunderstand newspaper hyped reportage of plasma therapy having worked in one patient, having worked in a second patient as real. None of those are properly controlled, soundly designed clinical trials, and therefore cannot be interpreted as successes. Are there plasma therapy trials going on? Yes, there are. But this is always going to remain a major problem with the practical implementation, even when plasma therapy is shown to be protective and licensed. When I take plasma from a donor, what tests will allow me to say how many milliliters of this plasma, this particular plasma, needs to be given in order to mediate protection? Until those tests are validated, you're not going to see plasma therapy. So, lab-based antibody tests can be scaled to commercial equivalents. That's actually really quite easy and not difficult at all. It's subject to normal manufacturing glitches and faults, but beyond that, there is really no major problem. How successful is soluble ACE2 protein in a human body? That's an interesting question. People are asking it. I know people who are trying to use recombinant ACE2 that's tagged to immunoglobulin FC portion as a therapeutic intervention. And I've seen one bioRxiv preprint report on it. So what we will wait and see. Keep in mind that that version of ACE2 will also bind to angiotensin, which may have its own consequences in terms of therapy. So nothing is not, things are not straightforward, because ACE2 is expressed on cells for a physiological purpose, which is likely to get disrupted if we mess with it too much. What's the difference between SARS-CoV-1 and SARS-CoV-2, and can vaccine be, SARS-CoV-2 vaccine doesn't really work well against SARS-CoV-2. That's something that people working on macaque models have shown. Opening of educational institutions, and the one question that had not yet come up of, is this a natural virus or is it a bioweapon, are the two questions that I will address. For everybody else whose questions I have overlooked, please forgive me, but really, we've been going for over 2 hours now. So let me answer the educational institution question. And the opening of educational institutions needs to be done with a great deal of calibration. I'm not suggesting that we will not be careful. All of us will be. I'm simply arguing for calibration of one step at a time. We need educational institutions to be open. There are student lives and livelihoods at stake. We are not a society, unlike many prosperous economies. We are not a prosperous economy. Everybody does not have access to fiber broadband and appropriate end-to-end technologies. So shifting, for example, for the entire next academic year to an online format, I really think would be unjust in our socioeconomic circumstances. We really do need to begin to teach in ways that do not exacerbate our socioeconomic inequalities. We do need to be safer. We do need to maintain physical distances. We do need to fall into cultural habit patterns that allow us to reduce, as I keep saying, the R0. We still do need to find ways of getting back into productive life that includes socioeconomic, that includes educational institutions. And I would have argued that individual educational institutions and the student communities should be talking to each other about this, about exactly how to configure and calibrate reopening. Because this is going to depend in part on where we are located. You know, being located in Nanded is going to be different from being located in Pune, is still going to be different from being located where you people are in Mumbai. Number one. Number two, what our class structure is like, what our student community socioeconomic distribution is like, what the college and university technological resources are like. It's going to be driven by all of this. And therefore, I would very much that in a common framework of care, caution, and evidence-based awareness, with the spirit of social solidarity, we approach this together about how to configure our coming back to a sustainable structure of teaching, of education, of socioeconomic life. I'm going to stop that there. The last question I'm going to talk about is also the most amusing one, which is the bioterror. Again, I will end, as it turns out, with what I began with, that when people in responsible chairs make irresponsible statements, the statements and the ideas acquire inappropriate prominence in life. And as I pointed out with hydroxychloroquine at the beginning, this one also is at the door of the responsible position of the President of the United States, making what appear to be responsible statements. So, is there any reason to believe that the virus could be lab-made? Forget about is lab-made. Could be lab-made. And every viral isolate sequence analysis that I have seen so far, the last time I looked for this was 3 days ago, every one of those analyses say there is no sequence-based information that we can see that says this may not be natural. There are bat coronaviruses. We know bats are major coronavirus reservoirs for a variety of remarkably interesting and speculative biological, immunobiological reasons. We know that civets have coronaviruses. We know pangolins have coronaviruses. We know that all of this zoonotic leaps can happen. We know that increasing human footprint on the biosphere is likely to increase the probability of zoonotic leaps of microbial infections of all sorts. Everything we know about SARS-CoV-2 today fits that natural framework and circumstance. There is no reason at the moment for us to join the conspiracy theory world that, unfortunately, some politics in the world is attempting to propagate as xenophobia. Let us not give in to xenophobia. Let us keep ourselves inclusive. Let us maintain our social solidarity, and let us deal with this as rational thinking people. Thank you very much.
Thank you very much, Satyajit. Before we wind up, Dr. Hemlata will give a formal vote of thanks.
Oh my goodness. Okay.
Thank you, Dr. Satyajit. It was a very extensive talk. I mean, I never thought it would continue on and on for 2 hours, but I'm sure our interactive session that you came up with was wonderful. And our students, I think in the coming days, as and when we get more and more information, will start thinking logically. We'll even start reading up scientific article, and I'm sure some may even zoom into your email ID and ask you some questions. So it was very good that we could organize this with you, and also special thanks to our principal and the DBT Star College team for making us think about this, and also our core team of members, Rinki, Shreyasi, Michelle, and Krishnendu. You two have done a wonderful job. So thank you very much, and we are sure that in the coming days, with 30 team in India trying to come up with a vaccine, soon we will be also hearing that information. And in the meanwhile, I'm sure the students have seen from you that there is a social connect that you have made, the socio-economic and international and community feeling that one needs to develop in the event of this pandemic. Thank you very much.
Thank you very much. Thank you all. I hope everybody got something, at least a little bit out of it. Thank you.
Yeah, thank you so much.